in increasing the fertility of the soil. Okay, so Aditya, you can start now.
Okay. Ma'am, as today the basic concern is pollution, and so control it.
Yes,
We have to use biofertilizers because the chemical fertilizers also harm the um, fertility of the soil, and it's also create pollution, and sometimes uh, if it can be dangerous for the living beings of the seas and oceans, water for the water bodies.
very
So
good.
biofertilizers are the organisms that enrich the nutrient quality of the soil,
Okay,
as
yes,
chemical fertilizers enrich, but they also harm the land or the soil, and but they have adverse effects later on. At the time of when we use them, they give the results are very good, but later on it decreases the fertility. And
yes,
but because this is a basic concern, now some companies also start selling biofertilizers that can be made from organic items, that can be cow dung and different items.
yes,
But as some microbes also helpful in them, they are bacteria, fungi, and cyanobacteria. So first we'll mainly talk about rhizobium, as we have studies in our in 11th only, and in 10th we are studying from starting about rhizobium bacteria,
yes.
that we get in the leguminous plants, which helps them to fix the atmospheric nitrogen that can be absorbed by the leguminous plants, so that they can absorb bacteria, so that they can absorb nitrogen in in free living in the soil, in free living form in the soil, so thus enriching the nitrogen content of the soil. And fungi also, some fungi have symbiotic relationship with the roots of the higher plants, that is mycorrhiza. So which have the genus glomus. So this fungal symbiote is in these association absorbs phosphorus from the soil and passes it to the plant. So mainly this symbiotic relationship help the plant so that it can get the phosphorus, and thus use it in a better efficient way.
Okay, very good.
Plant having such associations also show some benefits. The number one benefit is that such as resistance to root-borne pathogens.
Very good.
Second is tolerance to salinity and drought.
Very
Third
good.
is third is overall increase in the plant growth and development. So
Yeah.
these symbiotic relationships benefit the plants in a better way. Then the last one we have is the cyanobacteria.
Yes.
Cyanobacteria is mainly present in the oceans, and cyanobacteria are the cyanobacteria is mainly helpful in converting the producing 80% of the oxygen in the atmosphere, as our principal sir told us in the 11th class.
Okay.
So cyanobacteria are the autotrophic microbes widely distributed in aquatic and terrestrial environment, mainly of which can fix atmospheric nitrogen.
Yes, yes.
So in paddy fields, cyanobacteria is used which increase the salinity, which increase the fertility of the soil. Currently in our country, a number of biofertilizers are available.
Yes.
So we have to study about these three uh, microbes. That is, first one is bacteria, that is rhizobium. Second
Yes.
is fungi, that is mycorrhiza, that is symbiotic relationship. And third one is cyanobacteria.
Okay.
And
So,
also blue green algae, which add organic matter to the soil and increase fertility.
okay. So
Then
cyanobacteria is only the blue green algae you can call. And I want Aditya that the rhizobium, so it is symbiotic bacteria. You have read some free living bacteria also which are used as the biofertilizer.
no, ma'am.
Okay, anyone else if you have read in the textbook? Any
Ma'am, azoosperillum
azospirillum? Yes.
and azotobacter.
Ah, very good. So remember everyone, when you are talking about bacteria used as the biofertilizer, you have to remember that there are some free living bacteria also, and there are the symbiotic one. So symbiotic is the rhizobium, and free living ones, they also help in increasing the fertility by increasing the nitrogen content in the soil, and they are azospirillum and azotobacter. Okay, and then mycorrhiza, very well explained by Aditya. And cyanobacteria, also I want anyone to name the genus of the cyanobacteria. Aditya, you have read it? What are the different examples of the cyanobacteria used in biofertilizer?
Yes, sir.
What are they?
Ma'am, Anaebina,
Huh?
Nostoc,
Yes,
and Oxylatoria.
yes. So everyone will remember these names. One is Anabina, Nostoc, and Oscillatory. What we say? Anabina, Nostoc, and Oscillatoria. So Anjali, can you name these three?
Yes, ma'am.
Name it now.
Anabina, Nostoc,
Yes.
Osli,
Latoria.
Oscillatoria.
Oscillatoria. Okay, very good. So this way, I hope the biofertilizer topic is prepared by everyone and all have understood. So with this, we have completed with the chapter, children. Now we will be starting with the unit number 1. Okay, so we have completed with unit 5 and we have completed with unit 3. So everyone will try to finish up each and every topic of these two units thoroughly. And this way you will prepare how much marks? Guess you
Ma'am,
say
26.
26. ठीक है, 26 मार्क्स का सबका तैयार हो जाना चाहिए. दो दो चैप्टर दोनों यूनिट में है, so it's not difficult also, and they are very easy units, I think. All topics are clear to everyone.
Yes, ma'am.
Okay, everyone, so you can write in the chat box yes, and then we will start with the unit number 1. Okay, Manjul has understood. Okay, Shifa, Aditya, Yuvesh. Okay, Anjali, Nandini, Pranjal. Okay, so everyone will be able to prepare now the 4 chapters thoroughly. So today we will start with unit number 1. You can write in your notebook now. Unit number 1, and you know that first chapter of this unit has been deleted. We will start with the second chapter, that is reproduction, that is sexual reproduction in flowering plants. You can write down the name of the chapter, sexual reproduction in flowering plants. Ready everyone?
मैम ये वाला चैप्टर थोड़ा सा कर लिया था ना हमने?
मैंने पढ़ लिया, कितना पढ़ लिया?
यस मैम. थोड़ा बहुत कम ही हुआ था उसमें मैम, यही फिर क्वारंटाइन चालू हो गया था.
अच्छा, फिर से बोलो, आस्था अगेन.
मैम वही 2 दिन में थोड़ा सा कर लिया था मैम इस चैप्टर का.
तो अपन ने वो फ्लावर बस किया था ना, फ्लावर स्ट्रक्चर.
हाँ हाँ यस मैम.
हाँ ठीक है, तो वील जस्ट डू द रिविजन ऑफ इट और हम लोग आगे बढ़ जाते हैं. ठीक है,
यस.
तो एवरीवन इज रिमेम्बरिंग. We discussed that the flowering plants reproduce by the organ, that is the flower. So all the flowering plants, that is the angiosperms, have the reproductive structure, that is the flower. They all bear flowers, and the flower is the part which is having the male reproductive organs and the female reproductive organs. So I want someone to explain the different parts of the flower. I'm sharing my screen. So who will explain?
Ma'am, ma'am, I.
Okay. Poonam is ready for explaining. Okay,
Yes,
I'll
yes,
just
ma'am.
keep the
Ma'am, ma'am, can I
just
start?
I'm keeping the figure in front of you, and then you can start with it.
Yes, ma'am.
Okay,
Um,
everyone can see the figure. Screen is visible to you all.
yes, ma'am.
Okay. Okay, so you can start now.
Yes, ma'am. Ma'am, flower has four walls.
Yes,
Ma'am, first is calyx, which is made up of sepal. It is green in color and it protects the flower in bird condition.
very good.
And ma'am, second is corolla. It is made up of it is made up of petals, and it is very colorful and it attracts insects for pollination.
Very good.
And third is androecium. It is made up of stamen. It consists of
Yeah.
anther and filament, and anther lobe contains pollen grain. And fourth is gynoecium, which is made up of pistil. It consists of ovary, style, and stigma.
Correct. Very good. So this way, these are the four different words of a flower. Now, which are the essential words of a flower? Now, in this, which one you call it essential words directly involved in reproduction process?
Ma'am, gynoecium and androecium.
Very good. Gynoecium and endoecium, because endoecium is the male part and gynoecium is the female part. Okay. Now, so we call it essential words, and which are considered as non-essential words.
Ma'am, calyx and corolla.
So you have seen that they are not directly involved in the process of reproduction. Now, try to remember that the flowers in the different plants, we find they are of two types. We call it as unisexual and bisexual. Can someone tell me what is unisexual? What is bisexual?
Ma'am,
Huh? Yes, Astha?
unisexual has either the androecium or the gynoecium, but bisexual has the both androecium and gynoecium.
Very good. Okay. So now, who will explain me the structure of the anther? Anther structure, anyone will explain? That is the stamen structure. Anyone? Stamen structure. What are the parts of the stamen? Manjul?
Yes, ma'am.
What are they?
Ma'am, answer filament.
Anther and filament. Okay. So you can see on the screen it is showing that there are two parts of the stamen. It has got a long and slender filament. जो stalk जैसा structure है, that figure में भी अपन को दिख रहा है, that is the filament, and on top of it we find the lobed structure is attached, and that is known as the anther. So anther we find it may be bilobed and dithecous. Okay, and then anthers are attached to the filament in different ways. So, and the anther, when you see the TS of it, when you cut the cross section of it, you will find that it is having each of this lobe has got, तो ये एक लोब हो गया, ये एक लोब हो गया, so we call it as bilobed. okay, or and diathecus. That is two compartments are there. And each of the compartment we are finding, can you see this structure, the circular structures? This is known as microsporangia. What is it known as?
Microsporangia.
हाँ, speak up. What is
Micro.
it called, Aditya?
Microsporangia.
हाँ, and अपन एक को microsporangium बोलेंगे. Plural will be microsporangia. So you can find inside it how many microsporangia are visible in that one anther.
4.
Four are visible. Okay, so it is covered by the epidermis. Then you find the middle layers are there, and then in the inner layer, which is known as endothecium. Okay, so this way, this is the structure. This figure you'll get it in the textbook also, and everyone will have to draw it in the notebook. So all of you all draw the flower, that is LS of the flower also. Draw the stamen diagram and draw this microsporangia. Okay, that is CS of an anther. Now, inside this, I'll take up another figure. The parenchymatous tissues are present, and we call it as porogenous tissue. Each of this microsporangium contains what? What it
Porogenous
contains?
tissue.
Poro.
Pollen sacs.
हाँ, ये पोलन सैक तो ये पूरा हो गया. ठीक है, ये पूरा 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 आंसर होता है. आंसर ऐसे स्टार्ट हो गए. 4 स्टेप के अंदर एक एक लोब में क्या प्रेजेंट है? माइक्रोस्पोरेंजियम प्रेजेंट है. एंड इनसाइड द माइक्रोस्पोरेंजियम we find the parenchymatous tissue, which is known as porogenous tissue. Okay, and these porogenous tissues consists of the microspore mother cells. कि ईच माइक्रोस्पोरेंजियम जब मच्योर हो जाएगा तब वो पोलन सैक कहलाने लगेगा. अभी इनके अंदर पोलन ग्रेन्स नहीं है. जब तक पोलन ग्रेन नहीं बनेंगे तो अपन उसको पोलन सैक नहीं बोलेंगे. ओके, आफ्टर maturity then you will call it as pollen sacs. So which cells will make the pollen grains? Can anyone tell?
Ma'am, homogeneous cell.
Huh?
Homogeneous cell.
Ma'am, microspore mother cells.
Remember that. Okay, microspore mother cells will form the pollen grains. Now we will have to understand that how this process goes on. So the process of formation of the pollen grain is known as microsporogenesis. All have understood what is microsporangium, what is porogenous tissue, and what is microspore mother cell. Clear to everyone or not?
Yes, ma'am.
This much everyone understood. Now we'll see the next figure. So try to see here. अब इसी को एक enlarge करके दिखाया है. This innermost is known as the endothecium. Okay, और फिर इनके बीच में vascular bundle is present. So it is covered by each of the microsporangia. Remember the structure. I will be asking that describe the structure of microsporangium. So microsporangium is covered by three layers. That is epidermis, endothecium, and middle layer. And they all are protective in function. The innermost layer is the tapetum. What is the innermost layer known as? Tapetum. So can you see here it is marked. A tapetum के अंदर फिर parenchymatous cells है. That is parenchymatous tissues will be there. So what is tapetum? The innermost layer is the tapetum. So how many layers you remember? एक कवरिंग है. वो कवरिंग कितने लेयर्स का बना हुआ है?
3 layers.
3 layers. What is that? What are those 3 layers?
Outer one is epidermis, then the
Outermost
epidermis,
एपिडर्मिस. हाँ,
then the
फिर
middle
बीच
one is
वाले लेयर्स को अपन
endothelium.
मिडिल लेयर की बोल रहे हैं. फिर अंदर वाले लेयर को
Tapetum.
नहीं नहीं, उसके पहले एंडोथीशियम बोलना है.
Yeah,
व्हाट
ma'am,
इज दैट?
middle one is endothecium and the inner one is tapetum.
नहीं नहीं, सबसे बाहर एपिडर्मिस, फिर आए मिडिल लेयर्स, फिर आया एंडोथीशियम, ओके, फिर एंडोथीशियम के भी अंदर जो कवर करता है स्पोरोजीनस टिश्यू को, दैट इज नोन एज द टैपेटम. So tapetum is nutritive in function, and tapetal cells, they are binucleate or even multinucleate. Did you understand everyone? This much clear to everyone? And the young anther contains a group of compactly arranged microspore mother cells that is only known as the sporogenous tissue. Did you get it? And that is present in the center of this microsporangium. So clear this much to everyone. Shall I take up the next now slide? Noted all of you
Yes,
all?
ma'am.
Okay. The structure of the microsporangium very clear.
Yes, ma'am.
Okay. Alima will tell quickly. Alima.
मैम उसका मैम बहुत पुअर कनेक्शन है, तो मैम उसकी
अच्छा
मीटिंग बार बार
अच्छा.
लीव हो रही थी.
तो फिर who will explain? Who is ready? Nandini is ready.
Yes, ma'am.
Explain कर दो जल्दी से.
When microsporangium has four layers.
Okay, ठीक है. It is circular in shape and it
It
is
is circular,
covered by cells. ठीक है.
four layers. The first layer is epidermis,
हाँ
and middle layer,
हाँ.
and endothecium,
Endothelium. Okay.
and most inner layer is tapetum.
हाँ, तो फिर in this diagram you can understand that this one is your tapetum, and then this encloses inside it what? Then inside it what will be present?
Ma'am, tissues.
Sporogenous tissue.
Spor-
Inside it what is present? Sporogenous tissue. And sporogenous tissue contains what? Microspore mother cell.
Mother cells.
Microspore cells.
All learned this much? Yes or
Yes, ma'am.
no?
Ma'am,
Okay.
Yes.
Taking up the next. Now you have to understand the process of formation of the pollen grain. That is microsporogenesis. What is microsporogenesis, Manjul?
Ma'am, uh, formation of microspore from pollen mother cells.
Very good. Okay. That is microspore mother cell will form the pollen grains. That is the microspores. Pollen grain को ही अपन microspore भी बोलते हैं. And that process is known as microsporogenesis. So you can write down that within the maturing anther, the cells of the sporogenous tissue divide by meiosis. So what will happen? Each of the mother cell. So can you see here? This is one microspore mother cell. This microspore mother cell will undergo meiosis. So you know meiosis involves two divisions. So that's why here it is mentioning meiosis 1. So two cells will be formed. Then at the second meiotic division will take place. So four haploid cells are formed. And these four haploid cells will then be covered by the hard covering. Okay, that we call it as the. Are you remembering the enzyme and the in time? We did in 10th class. आई डोंट नो नाउ यू रिमेम्बर और नॉट. मतलब इस पोलन ग्रेन के एक एक के चारों तरफ हार्ड कवरिंग बन जाएगा एंड देन इट विल बी फॉर्मिंग अ मैच्योर पोलन ग्रेन. And the material needed or the substances needed for the formation of this covering of the pollen grain will be provided by the tapetal cells. So we are calling the tapetum layer as the nutritive layer. Did you get it? Did you see in the previous slide all of you are remembering?
Yes, ma'am.
So why we are calling it as the nutritive layer or it is made up of the nutritive cells? क्योंकि जो substances इसके wall formation में चाहिए होंगे that all will be provided by the tapetal cells. Okay, and so this way capital cells will be used up. And then you'll find mature pollen grain will be formed. Understood? So you find in different species, different shapes and sizes and colors will be there of the pollen grains. So did you understand the process? So this way, can you see this cross section? तो ये चार इसमें अपन को क्या दिख रहा है? Microsporangium. Inside each microsporangium, this process will occur, and this process will be occurring in which cells? The pollen mother cell or microspore mother cell, which is diploid. It will undergo meiosis, it will form four microspores, and then they will mature, they will be covered by the hard covering, that is exine and intine, and it will form the pollen grains. Clear this much? Shall I take up the next slide?
Yes, ma'am.
Okay. Now, we'll try to understand the structure of the pollen grain. Now, everyone's duty is that the figure which is given in your textbook, you will be drawing. First, the flower figure, then you will draw the a typical stamen given there in your book. And we'll make the figure number 2.3. You can note down. The figure number 2.3 is explaining the process of microsporogenesis. Okay, that is formation of the pollen. And once the pollen will be formed, remember जो अपन को चार दिख रहे थे अलग अलग कंपार्टमेंट जो ग्लोब्स दिख रहे थे there the cross wall will dissolve and ultimately the anther will have only two lobes. कितने उसमें थीका बचेंगे? दो. Beach का wall dissolve हो जाता है and then it will be filled up on maturity. I'm talking when it will mature. Okay, so that way the figure is drawn. You will make that A, B, C figure. Okay, now we are discussing what is the structure of the pollen grain. Each pollen grain, try to see here, it is covered by this hard covering. This hard covering is made up of two layers. The outer wall we call it as exine, and this exine is made up of the sporopollenin. Exine is made up of sporopollenin, and remember it is the most hardy and disease resistant organic material known. Now try to understand what is the need of it. Pollen grains are released from the anther and they are carried to distant places and they have to fall on the stigma of other plants. अब वो कहाँ से कहाँ कितने दूर दूर तक कैरी हो जाते हैं तो देयर आर चांसेस ऑफ गेटिंग डिस्ट्रॉयड so they are covered by
Exine,
the
Exine.
enzyme which is made up of.
sporopollenin.
Now, remember this word very nicely. Exine made up of sporopollenin, the most hard and disease resistant organic material known. Okay, most hard and disease resistant organic material known. And because of that, pollen grains do not get destroyed. वो कितने भी दूर चले जाए उनको इंसेक्ट्स लेके जाए बर्ड्स लेके जाए और the that is your bees and all they take it they all the pollen grains will not be destroyed. Understood? Then the exine has aperture. So this should be known that pollen on germination produces the pollen tube. Up from where the pollen tube will come out if the sporopollenin will be present in this whole structure क्योंकि वो तो बहुत हार्ड सब्सटेंस है तो this exine is having the aperture. Okay, and we call it as germ pores. These one or two points now the sporopollenin will not be present and that is the weak place there. So when the pollen grain will germinate, the pollen tube will arise from the germ pore through that germ pore. Is it very clear? Understood everyone?
Yes, ma'am.
Okay. Pranjal will tell me now quickly the structure that is what is exine. Say soon.
Ma'am, it is the outermost wall of
Huh?
a sporopollen,
Made up of sporopollen. Yes. Huh? Then
which is most hardly clear, and it's a diseases resistance organic matter.
material. Okay. And what else then you have to say what it has?
It's
Manjul, what
exine.
it has?
Ma'am, exine contain germ pores through
Germ
which
pore.
pollen tube emerge from the pollen grain germinates.
Sorry. Okay. During germination, the pollen tube will emerge through these germ pores. तो उधर से ही क्यों इमर्ज होता है? Can anyone give the answer? Astha,
Yes, ma'am.
why the pollen tube emerges through the germ pore only?
Ma'am,
Yeah, let Astha answer, then you answer. Sorry.
Ma'am,
Astha.
maybe because the hard cover is not present.
Maybe नहीं 100% that only.
Ma'am, I
Okay. Because the sporopollen is not present there. Yes, Aditya, can you explain now?
no, ma'am. I'm just asking the question.
Ma'am, due to
Say that.
presence of germ cell.
Due to the presence of नहीं बोलना है, due to the absence of sporopollen.
Yes.
Okay, at that place germ pores मतलब एक छोटा सा एक पोर जैसा स्ट्रक्चर है जहां पर स्पोरोपोलिन नहीं प्रेजेंट रहता है, which is the hard substance. So
Yes, ma'am.
that's why it arises from there only. Now in time, so inner wall is the in time, and that is thin, delicate, and it is made up of cellulose and pectin. Clear to everyone? Now try to understand the structure, inner structure, that inside what it has. So two coverings are there, exine and intine. Now inside the intine, each of the mature pollen grain you find will be having two cells. One we call it as vegetative cell, and vegetative cell will have the reserve food and irregular nucleus, large irregular nucleus. And the other cell is known as a generative cell, which is having a dense cytoplasm and a nucleus. So you remember that most of the angiosperms they release the pollen grains at this binucleate stage, means two cell stage में एक vegetative cell और एक generative cell. Okay, and in some cases what happens, the generative cell, so try to see here, this is your binucleated pollen grain and this is the trinucleate. This one is which cell? This is your vegetative cell. Vegetative cell में large nucleus है, and generative cell is having dense cytoplasm and a small nucleus. Okay, now in some cases what happens, vegetative cell तो जैसा है वैसा ही रहेगा, लेकिन generative cell divides into two, and when it will divide into two, तो you can understand there will be two nuclei. ओके, आप पूरे सेल को देखेंगे तो उसमें कितने न्यूक्लियस हो गए? In this case, how many nuclei? Can you see
see.
the figure and my cursor?
पहले मैं वो पीरियड बोल रही हूँ किसी क्लास में बोल
Smita?
I'm three.
Three, yes. So someone is keeping the mic on and some disturbance is there. Everyone listening carefully or not? You are noting the things or not?
Yes, ma'am.
Yes, ma'am.
Everyone has to note down. This is what you have to explain if it is asked. That describe the structure of the pollen grain. So you will draw the diagram as well as you'll have to explain the whole structure. So the pollen grain contains two cells, vegetative cell, I'm repeating now, vegetative cell and the generative cell. In most of the angiosperms, the pollen grain will be released in this two cell stage only, that is the binucleate stage only. In some cases, you'll find the generative cell will divide and it will divide into two nuclei. And then It will be in the three, that is your, how many cells will be there? Three cell stage and three nuclei. And then it will be released from the anther. Clear to you, everyone?
Ma'am, can you repeat again?
I am repeating the whole structure, and everyone has to learn that much. The pollen grain is covered by two coverings, or hard covering, which is made up of two coverings or two layers, you can say. The outer exile, which is made up of sporopollen, which is very hard and disease resistant organic material. It has one or two apertures present where sporopollen is absent, and these apertures are called germ pores. And during pollen germination, the pollen tube emerges through these germ pores. This much clear?
Yes, ma'am.
Clear, clear now, everyone?
Yes, ma'am.
Okay.
Yes, ma'am.
Now the inner layer. So inner layer is called, or inner wall is called intine. It is thin and delicate, and it is made up of cellulose and pectin. Now inside the covering, you find the pollen grain has two cells, one large vegetative cell and a small generative cell. And in that stage, so vegetative cell is having the reserve food, and it is having the large irregular shaped nucleus. And generative cell is having dense cytoplasm and a small nucleus. Now you will find जब anther mature होके dehiscence होगा, तो the in most of the plants the anther will be, that is your pollen grain will be released in this stage only, मतलब two cell stage में. But in some cases, you will find that the generative cell divides and then it forms three cells. मतलब दो male gamete बन जाते हैं. This nucleus, remember, these are only the two male nuclei, which you call it as first male nucleus and the second male nucleus. Are you remembering during the double fertilization we have discussed?
Yes, ma'am.
हाँ, तो इस तरह से वो दो पहले बन जाते हैं, और जिनमें अगर ये दो सेल स्टेज में ही रिलीज हो जाता, तो याद रखो जनरेटिव सेल डिवाइड कब होता है? व्हेन इट रीचेस द स्टिग्मा. व्हेन इट रीचेस द
Stigma.
स्टिग्मा. ओके, वहां जाने के बाद फिर डिवाइड होगा. Followed, but it has to have, the pollen grain has to have the two male cells, that is two male nuclei. Okay, all angiosperms have got two male nuclei. In some, you find that during the dehiscence and dispersal release only, they will be in three cell stage. In some, they'll reach the three cell stage after reaching the stigma. Clear to everyone? Yes or
Yes,
no?
ma'am. Yes, ma'am.
Okay. Now quickly we'll discuss the gynoecium. So this carpel constitutes the gynoecium. You also call it as pistil. So it is having three parts. The lower swollen part, which is known as the ovary, then it is having a long stalk, which is known as style, and on top it has got a receptacle, which is known as the stigma. So stigma, if you see from top, it is stigma, style, and ovary. And ovarian cavity, now this, when you see the ovary inside it, you will find it has got a placenta. And the placenta will have the, to the placenta attached the ovules. So the placenta gives rise to the megasporangia called ovules. So monocarpellary ovary, multicarpellary ovary, syncarpus, epicarpus, these terms we have discussed last year. Are you remembering
Yes,
when we
ma'am.
were
Yes,
doing
ma'am.
the family description? We were discussing syncarpus, epicarpus, when the carpels are fused, so syncarpus. When carpels are free, so apocarpus. Okay, and if one carpel only, we call it monocarpellary. And if many carpels, so polycarpellary. For example, lady's finger, tomato, brinjal. Okay, they are having more than one carpel. So this structure, this all we don't have to discuss just now. This all we have done in 11th class. Okay, the structure of the megasporangium. So this we will discuss next time. It is already 45. That is structure of the ovule. Megasporangium is what?
Ovule.
What is megasporangium?
Ovule.
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 Ovule. Okay, everyone knows ovule. Ovule only gets transformed into seed. मालूम है ना सबको?
Yes, ma'am.
That ovule gets transformed into seed after the fertilization. So the ovule is known as megaspore. And what is microspore? What is microspore?
Pollen grain.
जल्दी से बोलो what is microspore?
Pollen grain.
Pollen grains.
Very good. Pollen grain. And pollen grain are formed inside formed inside
Microspore mother cells.
नहीं माइक्रोस्पोर मदर सेल बनेंगे लेकिन वो किसके अंदर है माइक्रोस्पोर मदर सेल
Netatum.
हाँ माइक्रोस्पोर मदर सेल इज प्रेजेंट वेयर
Ma'am, sporangious tissue.
microsporangium बोल रहा है it is present in the microsporangium. Okay, so microsporangium consists of the microspore mother cell and these mother cells will only form the microspores. एक mother cell कितने microspore बनाएगा? हाँ, quickly tell me. This will form
4.
four.
Four.
Remember, each mother cell, each microspore mother cell will form four microspores or four pollen grains. Clear this much? So now we'll take up the structure of the megasporangium next time. So I'm stopping the sharing.
Ma'am, then where is microspore mother cells are present?
Microspore mother cells are present inside the microsporangium and microsporangium जो अपन sporogenous tissue बोल रहे हैं वही बना देता है cells का tissue is made up of cells. Okay, so microspore that is your sporogenous tissue which we are calling it is made up of the microspore mother cell.
मैम
Okay,
Clear?
प्लाज्मा
आगे तो
yes,
क्लियर
ma'am.
हो गया है ना
Yes, ma'am.
और एक एंथर के अंदर 4 माइक्रोस्पोरेंजियम हो गए और हर माइक्रोस्पोरेंजियम के अंदर स्पोरोजीनस टिश्यू है और स्पोरोजीनस टिश्यू का हर सेल क्या है
Microspore mother cells.
हाँ माइक्रोस्पोर मदर सेल और हर माइक्रोस्पोर मदर सेल will form how many microspores
4.
4 microspores is this clear to everyone
Yes, ma'am.
okay so who will name quickly the coverings of the microsporangium
Ma'am, epidermis, outer covering,
हाँ very good
cell layer,
हाँ
then endothecium,
very good
innermost layer is tapetum.
tapetum okay and what is the role of the tapetum Manjul can you tell me what is the role of
मैम
tapetum
it act as a nutritive function. It provide nutrition.
हाँ तो फिर वो किसमें हेल्प करता है जस्ट नाउ आई टोल्ड यू आस्था
Yes, ma'am.
हाँ what it helps in the tapetum helps in what
Ma'am, it is nutritive in function.
so what it helps in why you are calling nutritive in function who can answer
Ma'am, it provide nutrients to pollen grain.
पोलन ग्रेन तो वो न्यूट्रिएंट्स कहाँ किस काम में चाहिए
मैम पोलन ग्रेन के मैचुरेशन के लिए.
मैचुरेशन है, तो मैचुरेशन में किस प्रोसेस में? एनीवन, आई टोल्ड यू
मैम
फॉर्मेशन ऑफ
माइक्रोजेनेसिस, मैम
Microgenesis.
माइक्रोस्पोरोजेनेसिस।
नहीं, वो पूरा मत बोलो, फॉर्मेशन ऑफ द वॉल लेयर्स, रिमेम्बर, ओके। मियोसिस होके वो तो बन गया, लेकिन चार वो सेल्स माइक्रोस्पोर्स तो बन गए, लेकिन उसके ऊपर की जो कवरिंग, हार्ड कवरिंग है, दैट विल बी फॉर्म्ड बाय द material substances provided by the capital cells. Is it clear? Capital cells are providing the nutrition, substances as well as the hormones for the growth. Clear to all? This question can be asked, that what is, why you are calling, or how does it perform the nutritive function. Very clear to everyone?
No, ma'am.
What Aditya?
Ma'am,
Yes, say.
ma'am, ma'am, तो मैम ये फॉर्मेशन ऑफ द वॉल, इट हेल्प्स इन द फॉर्मेशन ऑफ वॉल लेयर,
Yes, that is your
दैट इज
exine.
एक्सटाइन.
Exine and entine.
ओके
Okay.
मैम.
सब्सटेंसेस विल बी प्रोवाइडेड, क्योंकि वो सब्सटेंसेस कहाँ से आएंगे, तो वो कैपिटल सेल्स अपना डिसॉल्व हो जाते हैं, एंड दे विल प्रोवाइड मटेरियल, द सब्सटेंसेस, और साथ में ग्रोथ के लिए हॉर्मोन्स भी प्रोवाइड करते हैं। क्लियर?
ओके.
हाँ,
यस सर.
okay, so now I'm stopping recording. So female system, the megasporogenesis, we will start